So first of all, we need to acknowledge that inflammatory bowel diseases are diseases which are increasing in incidence and prevalence, not only European-wise, but globally seen. And among all these different chronic autoimmune diseases, inflammatory bowel diseases are the ones who are most steeply increasing. And this particularly also in a young and adolescent patient population and in kids as well. So therefore the diseases put a lot of burden on those young patients because the patients are young, the patients are chronically ill and are affected by a non-curable disease. And this poses a social economic burden to those patients. More recently, there was a survey by the European Self-Health Group, EFGA, which delineated in detail all this burden, like problems in partnerships, problems in access to work, problems to maintain the work, and so on. I think this is a very important point. First of all, these kind of consensus activity is a major achievement of the European Crohn's and Colitis Organization. We should be proud of it. But on the other side, we also need to acknowledge that there are no firm and robust guidelines on how to diagnose Crohn's disease and how to diagnose ulcerative colitis. Something like this has, for example, been established for rheumatoid arthritis, but nothing is in place for Crohn's disease and ulcerative colitis. And so the diagnosis of Crohn's disease and ulcerative colitis remains a puzzle, putting different puzzle stones together, so from endoscopy, histopathology, clinical symptoms, and sometimes the picture fits and sometimes we are not really know what the picture is telling us. Also based on the heterogeneity of ulcerative colitis and Crohn's disease. And something like a prospectively validated tool to diagnose Crohn's disease and ulcerative colitis is clearly missing. So these guidelines are derived from national representatives of Europe, not only EU. This is real geographic Europe, and therefore they are supposed to have a global implementation and impact. So first of all, we should be delighted to see so much interest of pharmaceutical industry nowadays to develop biologics for patients with Crohn's disease and ulcerative colitis. So we were starving anyhow over the recent years on a lack of opportunities. But now some news and some new agents are at the verge of getting access to the clinical routine. There are agents not only addressing TNF-alpha and blocking TNF-alpha. There are now agents close to being available, which addressing adhesion molecules and very specifically block the entry of inflammatory cells into the bowel. There are agents which are blocking transmitter molecules and which have shown impressive results in ulcerative colitis. So there's a whole array of different approaches tackling different pathomechanisms and therefore widen up the window of opportunities of this heterogeneity of pathomechanisms which are underlying ulcerative colitis and Crohn's disease. Of course, if we could develop something like an individualized, or to use a very prominently used term of personalized medicine, it would be favorable and should be support. But I think we are not still there. Nowadays we do see drugs which have a certain kind of gut selectivity. Obviously, the study results of these drugs are looking quite bright. On how they perform later on in clinical routine needs to be shown. But nevertheless, I think we're moving in this right direction. So the knowledge of genetic associations in inflammatory bowel disease is exploding. It's unbelievable what has been achieved over the recent few years in this regard. So there are new data showing that about 160 different genetic polymorphisms are associated with inflammatory bowel diseases, and this on a balanced basis for Crohn's disease and ulcerative colitis. There's a huge overlap in the genetic disposition of ulcerative colitis and Crohn's disease, but there is also an overlap with other chronic inflammatory disorders, and even with bacterial driven disorders. This is of key interest, and we might learn more about this better mechanistically wise. But we also have to say that by now we can explain only 8 to 10% of all cases with these kind of genetic testings, and therefore it's not that we're not yet there that we can take advantage of these findings in the clinical routine, neither diagnostic-wise nor therapeutically-wise. For this, I think we need also to learn more about the individual contribution of these genes to the better mechanism of the disease. Of course, some of these genes are quite prominently expressed in patients, but they are also to be found in healthy individuals. And therefore, to learn more about the pathophysiology and maybe about side effects which could arise if addressing these certain kind of pathways need to be further substantiated. So genetics is one aspect on which we have learned extensively more recently, but we have also learned that obviously these genetic factors could differ from region to region, or from continent to continent, or from ethnicity to ethnicity, and what applies for a Western European population not necessarily needs to apply for an Asian population as well. And this also is important in our understanding how different the makeup of IBD, even despite a phenotypic similarity, is between different genetic backgrounds and different continents and races. And this might also explain why some drugs might fly and work nicely in one population, but maybe is not so active in another group. But this is a field which we still further need to explore in more detail to understand on how IBD is made up between different genetic backgrounds.